Picture this. You're peering into a microscope, looking at a clinical sample, and you see absolutely nothing. Well, that is the fundamental problem in microbiology. Bacteria are essentially transparent. And, if we can't see what's making someone sick, we can't treat it. We desperately need a way to illuminate this hidden microscopic world.
Welcome to this explainer. Today, we're diving into a genuinely fascinating challenge. How exactly do we light up the microscopic world? We're going to explore the brilliant science behind microbiological staining techniques and see how completely transparent microbes are transformed into vivid, readable maps of biology and disease. Here's our roadmap for today. First, the art of bacterial visibility. Second, differentiating bacteria, gram staining. Third, spotlighting specific cell structures. Fourth, the negative space strategy. And finally, number 5, advanced and diagnostic stains.
All right, let's kick things off with Section 1, the art of bacterial visibility.
So, when we need a primary coat of paint just to get our first real look at a microbe, we use what's called a simple stain. These rely on just a single basic dye. The heavy hitters here are methylene blue, crystal violet, safranin, and basic fuchsin. Applying just one of these is like flipping a light switch. It gives the bacteria color so we can instantly observe their basic morphology, which just means their shape and size, and figure out their cell arrangement. Are they clumped in clusters, hanging out in chains? A simple stain answers that for us right away.
Moving right along to Section 2, differentiating bacteria, gram staining.
Okay, let's dive into this. Once we can actually see the bacteria, how on earth do we tell them apart? That's where we bring in differential staining techniques. These are methods that use 2 or more stains to differentiate between different types of bacteria. Using multiple stains is absolutely crucial. It's the big step that takes us from merely seeing a bacterial cell to actually categorizing and identifying it.
And this brings us to the absolute cornerstone of microbiology, the gram stain. Developed by Hans Christian Gram, this method is a real game changer because it broadly divides the bacterial world into 2 massive categories, gram-positive and gram-negative. So how does it actually differentiate them? Well, it all comes down to their cellular armor. Gram-positive bacteria have these incredibly thick peptidoglycan cell walls that trap the purple primary stain. Gram-negative bacteria, on the other hand, have very thin walls, so during the washing process they actually lose that purple color and are forced to take up a pink counterstain instead. It's this incredibly elegant way to instantly categorize an organism based on its physical properties. And honestly, it's exactly how doctors figure out which antibiotic might cure your infection.
But gram staining isn't our only differential tool. We also have acid-fast staining. Developed by Franz Zeil and Friedrich Nielsen, this technique hunts down specific, highly resistant threats. Whether we're using the standard Zeil-Nielsen stain or the modified Quignon method, this technique lets us break through and differentiate really tough bacteria that normal stains just bounce right off of. And I mean it when I say this specific stain is quite literally a lifesaver, because it's absolutely critical for rapidly diagnosing tuberculosis.
Which brings us to Section 3, spotlighting specific cell structures.
Now, sometimes we need to look beyond the whole cell and focus on specific accessories attached to the bacteria. And different structural accessories require their own unique spotlighting methods. We care about these structures because things like capsules and spores act like heavy biological armor, making the bacteria much harder for our immune systems to fight. For instance, to see a protective capsule around something like Klebsiella pneumonia, we might use Anthony stain, India ink, or nigrosin. If we're hunting for incredibly hardy bacterial spores, like those of Bacillus anthracis, we deploy the Schaefer-Fulton spore stain. And for flagella, those are the tiny whip-like tails bacteria like Salmonella enterica use to swim around. For those, we use specialized methods like Leifson or Gray.
And these structural stains can get incredibly precise. We can target the cell wall itself, or even look deep inside the cell for highly specific internal features. A great example is metachromatic granule staining, using the Albert method. This is specifically designed to detect volatin granules, which are basically little internal nutrient storage units inside organisms like Corynebacterium diphtheria. It's pretty much like having biological X-ray vision.
Let's jump into Section 4, the negative space strategy.
Now, what's really interesting about this slide is how negative staining takes a beautifully reversed approach. Instead of dyeing the bacteria itself, the background is stained, leaving the bacterial cell completely untouched. It's exactly like looking at bright stars against the dark night sky. You see the subject perfectly outlined by the darkness surrounding it.
The most common stains we use for this reverse technique are India ink and nigrosin. But why do we do it this way? Well, first off, it's absolutely incredible for capsule visualization, since those protective outer layers repel the dye, creating this really cool clear halo effect around the cell. Second, and this is a big one, because the dye never actually enters the cell, the bacteria don't shrink or distort. So this gives scientists highly accurate measurements of the bacterial size in its natural, pristine state.
And finally, Section 5, Advanced and Diagnostic Stains. First up in our advanced rapid-fire toolkit are fluorescent staining techniques. These use special fluorescent dyes and, as you'd guess, require a fluorescence microscope to observe. Techniques like the uramine-rhodamine stain for acid-fast bacteria or acridine orange literally make the target bacteria glow brightly against a dark background. This allows for amazingly rapid detection of specific pathogens, which, again, is vital for identifying things like Mycobacterium tuberculosis or when using targeted fluorescent antibodies in immunofluorescence.
Now, sometimes bacteria are so incredibly thin and spiraled, we call these spirochetes, that traditional dyes just wash right over them. We simply can't make them out. So scientists turned to silver. By utilizing silver staining techniques like the Worth and Starry stain, extremely slender pathogens like Treponema pallidum and Borrelia burgdorferi are rendered sharply visible. They go from being totally invisible threads to very dark, distinct spirals.
Rounding out our advanced techniques, we have special diagnostic stains. This includes the Gemza stain, the Wright stain, the Fontana-Masson stain, and the periodic acid Schiff, or PAS stain. We even expand our capabilities right into the realm of fungi with lactophenol cotton blue. These are highly specific diagnostic tools that leave absolutely no microscopic stone unturned. It's essentially the ultimate detective kit for modern pathologists.
So, the crucial point is this. Look at our complete microscopic toolkit. Simple stains like methylene blue give us that baseline morphology. Differential stains, especially the Gram stain, act as our ultimate categorizer based on cell wall thickness. Structural and negative stains pinpoint specific protective features like capsules or spores. And our advanced fluorescent and silver stains offer that rapid, highly specialized detection. The takeaway here, every single dye in this toolkit has a highly specific, vital purpose in mapping out the invisible world. Which leads us to a final thought to ponder today. If just a simple dye can reveal the hidden, complex architecture of disease, what else is lurking out there in the microscopic shadows, just waiting for the exact right color to bring it into the light? Thank you so much for exploring the art of bacterial visibility with me on this explainer. Keep asking questions and always keep looking closer.